So it is my pleasure to introduce Dr. Michele Mansfield. Dr. Mansfield is the director of the Penn State University Honey and Pollen Diagnostic Laboratory. She's going to be talking with us tonight about the services they offer and some of the science behind how they analyze the honey samples that we send in. I know a number of our members have already sent in samples, and there are a lot of us that are going to be sending in samples. So welcome, Michele. We're really thrilled to have you, and I'll turn it over to you. And Michele mentioned that during her presentation, if there are questions, you can definitely feel free to put them in the chat or ask them as she goes along through this. So thank you very much, Michele. It's all yours.
Oh, perfect. Thank you. Ian, thank you so much for having me virtually here this evening. I'm really glad to get the chance to come and talk to you guys. I'm gonna apologize in advance that hopefully like my children, my cats, like the only time I see either of them is if I'm trying to do something on the computer. But I'll do my best to work with it. And I am not very technologically advanced with the computer, so sharing my screen and things like that can sometimes be fraught with peril. So I'm gonna go ahead and share my screen. Hopefully it'll load. Okay. All right. Let's see. It's supposed to be showing my presentation, and I don't think it is. Okay, hold on. Give me 1 second.
Take your time.
Okay. All right. I might have to download the presentation very quickly. I thought it would actually work off of my main channel, but it doesn't seem like it's behaving. So my apologies for this might take just a second.
No worries. And if you're just joining us, we're going to be beginning in just a minute or two. Dr. Mansfield is just getting her PowerPoint working, and we're going to jump in here. If there are questions and things during the presentation, feel free to put them in the chat.
Okay, I think I've got it now. Okay, are you guys seeing my presentation?
Yes.
Okay, yay, woo! All right, challenge number 1 down. Okay, all right. So I'm just gonna go ahead and talk a little bit about how I got here. I am a plant pathologist by training, but a lot of that training wasn't just for discovering what kinds of things are killing people's crops. It actually came in handy with learning quite a few diagnostic methods, both molecular and chemical. And 5 years ago, Dr. Christina Grosinger's lab manager, Kate Anton, is a very good friend of mine. And she hilariously was like, hey, want to come in and like do some work for part-time and get away from your kid? Not that I don't love my child, but being a stay-at-home mom was 100% the hardest job I've ever had. So I kind of got the job by default, and then came to find out later that Christina was not just looking for someone to come in and do a little bit of work here and there. She was looking for someone to get a lab set up, which thankfully I have quite a bit of experience in that as well. But beekeeping was completely new to me. I don't keep bees. I go over and I will help Kate mostly by being in her way and getting stung. But I'm trying to get a little more real world experience. So I do volunteer as often as someone's willing to put up with my not expertise in actual hive management, just because I think it's important to understand people that I'm hoping to offer services for. And while I'm giving this talk, again, like as Ian said, please feel free to like interrupt me, ask questions. I prefer to have a dialogue rather than just a straight lecture. I think that it also makes it more interesting for me too, because I'm not gonna lie, not my first rodeo giving this talk. Okay, so a couple of things I'm gonna go over. At its very basic, what is DNA metabarcoding? Barcoding and metabarcoding are two kind of different things. Back in the old school day, I just learned them and called them sequencing, which was acquiring a DNA sequence of an organism. But barcoding is kind of like the sexy new term for that, which is kind of hilarious because having been out of rotation for about 5 years, when I did come back, I'm like, I now have to relearn some slang. That's interesting. But I'll talk about what metabarcoding is, a little bit about how specifically pollen metabarcoding is used, a little bit of overview of our pollen identification service and spotted lanternfly, which I really, really hope you guys never, ever, ever need to send honey in for that to be tested. Here's to hoping. And then I'll also show a little bit of, showcase some of the other resources that the Center for Pollinator Research offers, and talk about some of the new services that I'm hoping to offer sooner rather than later. Because right now we have 2 services, and I would like to become more comprehensive. Okay, so traditional pollen identification, some of you may know this, some of you may not. I always think it's worth bringing it up. So I know that there was a gentleman, a faculty at Texas A&M, that for years did this kind of work. And traditionally how pollen was identified in honey or in a collected pollen sample was that you took that honey, you concentrated it, you filtered it out, you graded it out over a screen, and then you very painstakingly look under a microscope and would identify each pollen grain and then count all of those grains that you're looking at. That sounds a lot harder than once you see what I'm doing, it sounds like it's way more complicated. And that's, to be honest with you, is more of an art even than a science. I mean, it's absolutely science as well, but it requires a lot of training. As the picture might show you, pollen granules look like they have a lot of ornamentation. This is a very stylized picture. Most people who do offer this service, they're similar in that they can probably really only get to the family level. There just aren't enough distinguishing characteristics to be able to distinguish at a genus level. There's certainly not enough to distinguish at a species level most of the time. It also requires many, many hours of training to get somebody who's accurate. It's extremely time consuming. You can't, you have you can only look at one sample for so long. You only have so long to process each sample. And how many could you realistically get done in a day? It also, again, like I said, it's, if you're somebody who's very experienced, you might be very quick, but if you're training someone, that's going to take time for them to achieve that level of skill and look at enough samples to become a flat out expert that quickly. I will be the first one to admit, embarrassingly enough, that when I look under a microscope for any period of time without taking Dramamine, it's not pretty for anybody. I found out that I actually definitely get motion sickness when I was TAing the class when I was in graduate school. It was not the time to show your students that you needed to run out of the room quickly. Part of this training and being a scientist, right? Okay, so moving on to DNA metabarcoding. So traditionally, you're going to see 2 different types of descriptions here. You're going to see DNA barcoding, which again, barcoding is just another name of saying you're sequencing something to interrogate its DNA to find out what it is. Okay. And then of course, I'm going to also discuss like how that compares to metabarcoding. In a traditional sample, what you're going to have is you're going to have one organism that's either a pure culture or a pure specimen of just that organism in that sample that you're looking at. This is required for traditional barcoding because you can't have mixed organisms in there or you're going to get an absolute garbage mess when you get your sequencing data. It's not going to make any sense. It's going to be all scattered up and mixed up together. Metabarcoding, on the other hand, takes DNA from an environmental source. So you'll often see this called eDNA. I'm old school, so I just refer to it as genomic DNA. eDNA would be the more proper terminology because when you're looking at an environmental sample, there's no expectation that it's going to be a pure culture, right? Of course not. It's a real world sample. And this can be river water, soil. And in our case, we're going to be talking about honey, specifically the pollen that's in honey. And in the end, you can actually take all the organisms and identify them simultaneously. And here's my analogy, and hopefully this makes sense to everyone. So Penn State's ginormous stadium, Penn State Beaver Stadium, holds 110,000 people. Maybe not that right now because it's undergoing construction, but imagine if you were standing at one of the entrances to the stadium and there was a line of people a mile long. In traditional barcoding, I would be individually greeting and identifying each person as they walked in. Meta barcoding, on the other hand, imagine if I'm standing on top of that stadium and I take a snapshot of everybody in that stadium and immediately I have everyone's identification. That's the difference and that's the power. You can basically interrogate a sample with a specific group of organisms in mind. Either maybe you're just looking at the plants, maybe you're looking at the bacteria, maybe you're looking at the fungi, but you can identify all those things in one shot in those samples. And that's why meta barcoding is so powerful, because again, traditionally in a sample, you'd have to tease apart every individual thing, isolate it, and then identify it, and then reassemble that back together somehow, which is artificial and it's obviously going to be prone to a lot of error. I wish this existed 20 years ago. My PhD would have not dragged on nearly as long as it did. This would have come very much in handy. Okay, so that's just a little bit of description about what metabarcoding is. So what does this mean for application, specifically to your group and your interests? So metabarcoding is great because I can identify any plant material that would be in a particular sample. It doesn't necessarily have to be strictly limited to honeybees. This can also, well, I've done studies with wild bees. This is usually with research groups, or basically any plant sample. My favorite so far has been someone was studying hoverflies. And I also was contacted by a group that's looking at hummingbirds. I would really want to come meet some hummingbirds and get some pollen samples. That just seems pretty awesome to me. Obviously, beekeepers, they want to know what their bees visit. And obviously, again, on more of a research level, a lot of this work goes to identify and drive conservation efforts. Particularly, I did work with a group that actually they were out of Fiji, which was really fun. I ended up identifying a plant that was invasive in one of their areas, and they were able to find it and rip it out, which I was like, at first I kind of freaked out. They're like, it shouldn't be there. And I'm like, I didn't make a mistake, I promise. And they're like, no, that's good that you told us, because now we can go back and rogue this weed that can become a real problem really quickly. So there are many applications to what this can be used for. Oh, skipped ahead too far. All right, so this was a really fun project that I worked on with a colleague of mine named Michele. It actually was really funny because we had to just distinguish. I'm Michele with one L, she's Michele too. And we would give talks with one another. I'm like, okay, which Michele are you talking to? So one of the projects I helped her out with was she had samples that she collected with several members of her lab in UC Santa Barbara, and they did visual observations on several species of bumblebee that they were working with in that general area. And these were the plants that they observed their bees going to. And I mean, these make sense, right? But the metabarcoding uncovered a whole other group of plants that they had no idea that, I mean, there's only so much manpower in the world you're gonna have. You're not gonna run out and chase down every bumblebee you see, right? My daughter would try it, but I don't recommend it if you're trying to put your time and effort in research. But it's interesting to see like how much more they were able to discover that the bees were actually visiting. So that's another really interesting application. And then something else that is kind of near and dear to my heart, and I'm hopefully going to be working with some groups in the future on this. There's a real excellent chance that we could really interrogate historical collections of bees and other insects that go after pollen. And some of the samples we have in our Frost Museum on campus are well over 100 years old. They've been fairly well preserved, and if we can get at the plant DNA, it would be really interesting to see how, if you had enough specimens from certain locales. I mean, this, by the way, is where I'm at right basically right where that arrow is. I'm about 10 miles north of my house from the university. But it's really interesting to go back and be like, how much has our landscape changed in the past 100 years? And a lot of these archive samples could provide a really, really, really interesting insight to that. I'm actually working with some folks, hopefully in Southern California, that want to look at this a little bit too, in collections across the world. So that would be like another more broad research-based application. Okay, all right. So now for the more specific to you guys application. Okay, so it should, most of you probably are already aware of this, but the service is for citizens as well as research scientists. So anybody who's got an interest in what they have around their bees, their honey, their pollen, whatever. I will accept both uncapped and commercial ready samples. I'll get into why occasionally I prefer just the scraped uncapped samples. Pollen from pollen traps. If any of you have a pollen trap and want to submit a pollen sample, if you can and have that ability, I strongly recommend that. I'll get into why that is also a little bit later on. And then again, like you saw in my previous slides, I'll also take pollinators, and hopefully we very, very carefully. It's actually a little bit funny watching adults with this teeny tiny insect, like very gently brushing off pollen. So yeah, talk about an interesting, like when my mom asked me what I do at work today, I'm like, well, I brushed some bees off. She's like, yeah, okay, great, honey. Okay, all right. So I don't know how many of you have received a sample collection kit. Hopefully they look much better than the one in my picture. This was literally kind of like a last minute thing, and I can get some better photos and update my talk at some point. So for sample collection, the kit consists of everything that you're gonna need. If you're collecting honey, I ask for two full tubes per sample. 1 tube if it's pollen. It doesn't necessarily have to be full if it's pollen, but usually I ask people, like, if you give me like up to like the little first tick mark, which is like roughly 5 grams. There's going to be a sample information sheet that comes with every that you should have as many information sheets as you have samples. If you don't, I always tell people, email me. I've got it. I also have it posted online on our website. You can always download it yourself. And then I do prepaid postage. You can send everything back when you collect it. And then the QR code should go to the website for the lab. And the website, I really strongly encourage people to go to the website and read. Whenever you contact me, by the way, like 99% of the emails, I'm the one answering them, and I'm training my technician to do that now. And yes, I do have form emails. So if I come off sounding a little formal, even though I've met you, I'm not trying to be that way, but I get a lot of requests on a weekly basis. Okay. So sample collection. For honey, again, I ask for 2 tubes per sample. This is for a couple reasons. Uncapped honey would be basically if you can take that tube and you scrape it across a frame with nectar or honey in it. That is the most ideal way if you're going to give me a honey sample. I can do consumer ready honey. There are a couple caveats to that, which I'll get into a little bit later. It can be done, but that's why I ask for 2 tubes. If it's a single, if it's a scraped sample, I really only need 1. If it is extracted sample, I ask for 2. Once I get the samples back at the lab, I warm them up. My daughter loves my crazy, it's not a water bath, it's a bath that has aluminum beads in it. So she's just riveted. I'm like, each of those beads is a fortune. Leave them alone and don't touch them. But I warm this in my lab is like really interesting to my 7-year-old right now. But the samples are heated. If there's wax in there, the wax comes right off. It's not a problem for what I'm doing. And then the sample, I take that 50 ml tube and I literally concentrate it down in this itty bitty teeny tiny tube. And believe it or not, that's actually one of the most laborious parts of dealing with honey is the concentration. It takes about 4 hours for me to go from 2 50 ml tubes down to like an itty bitty teeny bit of material. So for pollen, if you have a pollen trap, and actually if some of you could get back to me at some point, I've seen commercial pollen traps that our lab uses. They're like $100 a pop. They're pricey. I don't know what they're like, but I ran across on both Better Bee and some of the other commercial beekeeping sites and Amazon, they had pollen traps that were like $15, which immediately it's sort of like, oh, that'd be awesome if that was that cheap. But I'm also like, why is it that cheap? So if any of you have ever looked at seeing these pollen traps and have any experience with the expensive versions versus these El Cheapos, I'd love to find out about it, because I would like to keep some on hand for some of our local high school groups that I'm working with, because their programs really can't afford to put pollen traps on some of their hives. But it would, if the El Cheapos work, it would be nice to have an option that I could purchase for them and like loan them out. And actually for interested beekeepers too. But anyway, getting back to the topic at hand. So for pollen collection, usually the standard we recommend is you put a pollen trap on for 7 days. You collect it each day. You put it in a bag, you hand mix it as best as you can. You send it to me, and then I do something called homogenization, which is basically just you're taking all of it, mixing it together really, really well, and making sure that all the pollen, no matter where you take that sample from, would be equally dispersed throughout the whole sample. And obviously you can understand why this would be important. For pollinators, again, it's actually kind of hilarious to see a bunch of grown adults very gently brushing bees. When we get the pollinator, we bring it to the lab. We literally very gently will brush off either the pollen basket. Some of them actually need to brush, and some of these are teeny tiny. But we generally can collect enough pollen, and then we take that sample, and all the samples end up going in this fancy pants machine that I have pictured on here called a centrifuge. Again, it's noisy, it spins fast. It's very popular with the 7-year-old. Okay. But all of these steps are basically simply to take that sample and reduce it down to just getting at the pollen. Okay, so I'm going to try to not get too super techy here, but some of you might really be interested in the details, so I like to provide them when I can. Most of this process can be broken down into pretty finite sections. So obviously you're collecting the sample for me, I receive the sample, and I'm processing that sample to either make sure it's homogeneous or concentrating it to get at that pollen. Once I get it back in the lab and I've gotten to where it's concentrated, I've got the amount of pollen I need, we have this amazing machine that basically takes the pollen, and the pollen is collected from the honey or the pollen sample itself. And the tubes actually, they look just like this, but they have teeny tiny 1 millimeter chrome beads. And they go in this machine, and basically this shakes the snot out of them. I mean, again, this is the loudest, most obnoxious piece of equipment I have. And this is by far the 7-year-old's favorite machine, because it's super loud and kind of scary, actually, if you ask me. But the machine will basically, it's breaking up and what we would call lysing the cell. So you're breaking the cell open to get at the DNA, which is what I'm after, because that's what I'm going to use to identify the plants that are in the sample. We then use the commercial kits to purify that DNA away from the other cellular garbage that was in there that we don't need. It's just going to interfere. The DNA has to be quantified. So I need to know how much is in a sample. Believe it or not, one of the biggest problems I run into for pollen is that I have too much DNA, and I actually will have to dilute it with a little bit of water, because there's so much DNA that the amplification reaction, which is what I'll talk about next, it gets gummed up because there's too much DNA, which you would really think that that would be the opposite, but there's a lot of DNA in pollen. And then the last steps are kind of what I call like the techie geek side of it. So we use a process called PCR. You guys may be familiar with this if you got a COVID test and it was a PCR COVID test. PCR is essentially taking a target piece of DNA that you're interested in. In our case, I'm interested in ribosomal DNA because that's what I use to identify the plant. Taking that piece of DNA and photocopying it millions and bazillions times. So, oh, sorry. Go ahead. Does somebody have a question? No? Okay. All right. Well, I'm going to keep going then. So the PCR amplification, it's photocopying that target piece of DNA that I'm interested in. We have 3 rounds of PCR that make up our method. And basically they each do a different thing. The first round of photocopying is literally just making more of that target piece of DNA of whatever plants are in there. Okay. The second round is something that you're amplifying again, but you're actually adding little molecular tags to that DNA that tell my sequencing facility when they run their reaction, yes, this is good DNA. This is part of the whole group that needs to get sequenced. The last part, and this is what makes it meta barcoding, the last round of photocopying literally takes a tiny six base pair piece of DNA and incorporates that into that fragment. So I know what sample that DNA belonged to. It tells me that this sample belonged to whoever's name that sample reaction was. So when they go back and assemble it at the end, I know that that was whatever sample so-and-so sent me. And then the last step, because I know there's a lot of steps with this, once I've done my rounds of photocopying, I clean. Yeah, you have to clean everything, right? I know. Nothing, you can never, I feel like you're never going to escape from cleaning. But I clean that reaction up and I send it to a facility in Philadelphia that performs something called Illumina sequencing. Illumina sequencing essentially will take all that environmental DNA, amplify it one more time, and then pull down the information from that DNA, which they then send back to me. And I work a little bit of magic. I'm not a programmer, so learning how to use some of the data analysis side of things was a little bit challenging. But the final part of the process is that I get that DNA back and we run it through a data analysis pipeline. That data analysis pipeline does several things. It basically takes that information, discards anything that's not quality sequence, and then it takes that quality sequence and then compares it to a database that's got plant sequence from all over the world. So basically, if a plant has been sequenced, I have access to that information. And then that's where we get our identification. And not only will it tell me what the sequence is matched with, they will tell me what percentage of reads. No. I knew my kid was going to make an appearance. But it'll tell me what proportion of the sequence reads belong to what plant. So it kind of does many, many things at once. But in the end, what you'll end up getting is this fancy report that I will send you. And you can also see copies of this on the website if you go to the FAQ for the pollen identification. So all the reports include a total sample report. You would only get this if you submitted more than one sample. So if you submitted two, you'd get a total sample report, and then you would get your individual sample reports. And if any of you, when you get the samples, any of you, if you have ideas or feedback to make it more user-friendly, I'm all ears. I actually had a laugh. There's when I gave this talk 2 years ago, I'm not a programmer, so I had to learn how to generate the reports myself and do a lot of computer things that I was kind of nervous about at first, but it worked out. But I had to laugh because one of the commercial groups that offers DNA barcoding remodeled their reports to end up looking exactly like ours. I don't care. I mean, I'm not, but I just thought that was kind of hilarious. I was like, hey, no honor among thieves, right? But yeah, so if you guys think of anything that makes the report better, I'm all ears. We're always trying to improve how things, appeal to beekeepers to make it more user-friendly. Okay, so this would be an example of what a total sample report would look like. It drives me crazy that we haven't figured out a way to italicize the Latin name, just because, I know, I get fixated on stupid things. But what you'll see is you'll get a pie chart that lists the Latin genus name of your group of plants, and then we give a common name. This has caused some issues for me in the past, but I do feel it's important to provide some type of common name, but I've also learned that common names in Pennsylvania are not the common names you're going to have in California. So sometimes there's like a little bit of confusion, like, whoa, where did that come from? And I'll get into some of those kind of oddities later. And then we also provide an other column, because I'm not going to lie, you'll see a lot of others in there. And the others will be listed. So I mean, you can have a laundry list of other plants, and we do that because otherwise the pie chart would be pretty much like unusable, because you need a microscope to see all the little tiny people or tiny like separations. We used to have these called heat maps. I did end up taking them out because they seem to cause more confusion, but I like to show them occasionally when people have if they want to see the data represented in a different way. What this map is going to tell you is if you're looking at a particular plant across a number of samples, it's going to like basically like the darker and more intense the color, it kind of is just like a visual graphic of who was hot, who is not across a group of samples. And again, we don't offer this anymore on the reports unless we get a specific request to do it, just because we had a lot of people that were like, whoa, like, why are you, like, what does this mean? It kind of got a little too complicated. But again, I've are there other ways to represent the data? Yes, I do. And yes, I can. And I'll move along to what you're going to see in an individual sample report. Same kind of idea. I always make sure that every report has the information that you provided, because I've actually had some, I mean, we're human, and yes, I do sometimes give my technicians stuff to enter in, because I can only do so much on my time. So I like to make sure that people just validate that yes, their data, the information they give me in their sample sheet's correct. Same kind of idea though, you're going to see a proportion of what you're going to be seeing of a particular genus across your sample. So here are the things to keep in mind when you get this report. As I stated a little bit earlier, the common plant name is not representative. I actually had a kind of a hilarious question a couple weeks ago, because I didn't know this either. I had somebody that lived in the middle of, I think it was like they live in downtown Manhattan, and they're like, why am I getting like alfalfa in my sample? Alfalfa, the genus name for alfalfa is Medicago. And turns out, I didn't know this either, a super common like weed that grows between the cracks in the sidewalk, and yes, it flowers, that's where they were getting it. It wasn't actually alfalfa that they were seeing in their sample. We figured it out, because they're like, I literally have no idea where they're like, unless my bees are traveling statewide. So keep that in mind when you get that. I've had a lot of people be like, that doesn't grow in my yard. And I'm like, okay, it might not grow in your yard, but they can travel to 3 to 5 miles to forage too. So the other, hold on one second. Of course, my kid interrupts me. I'm giving a talk to a large group of people. Oh, okay, put that spike. Okay, great, thank you. Sorry, we had to stop and rescue lightning bug. Okay, my apologies. The second part of one of the second things I think it's important to remember, honeybees and native bees are generalists, so they will collect from a huge variety of plants. Plant DNA might not actually come just from pollen. So we also had kind of a strange phenomenon that we were running into. Again, this probably wouldn't apply so much to California, but we were ending up with conifer pollen, or not, we thought it was pollen at first, but we're like, is it blowing into their apiary? Like, what's this coming from? Turns out that you can pick up plant DNA from spotted lanternfly honey, because the honeydew that the spotted lanternfly feeds on, there's enough after it passes through the digestive system that my metabar cutting will occasionally pick it up. Because we were getting some really oddball, we were getting plants that we knew darn well their bloom time wasn't happening when it is now showing up in a particular sample. So that's kind of another oddball. And before I forget, the other thing I should mention about the common name not being completely representative, many plant genre, so a genus can have up to 600 species in it. So again, I give the common name, the one name that people would associate, but if you do see a genus that you look up and you're like, oh, I don't think this is in my area, don't look at the common name. We always direct people to go look at like iNaturalist or something like that and be like, verify that this is not really in your area. I mean, it didn't, and again, I'll talk to people about this too, if they have issues with it. One other thing that I have to bring up, so honeybees did not evolve in North America. A lot of the plants that we put in that are native, honeybees may actually not be able to collect pollen from them. It doesn't mean that they can't pick up the nectar, but if they're not collecting the pollen, it might be really difficult for me to be able to identify whether or not they visited that plant. Two really good examples of this are Monarda, also known as bee balm. Bee balm is buzz pollinated. Honeybees don't buzz or vibrate at the frequency necessary for that pollen to naturally be released. What's kind of also hilarious is when I watched a research presentation on this, they use electric toothbrushes to collect the pollen. I was like, that's a hilarious use for a super expensive electric toothbrush, which they don't, they bought like the kid versions. But another example of this is milkweed. Yes, I've got a beekeeper in PA that has a huge field of milkweed. And I firmly believe, and every once in a while I'll detect it in his honey, but most species of Asclepias milkweed, the pollen is sticky, so the bees will literally clean it off of themselves before they get back into the hive. So they're actively removing it, my ability to be able to detect it. So there are limitations. There are limitations. And again, knotweed makes a lot of nectar, but it doesn't often make pollen or very much pollen. So that can be a tricky one. And that's one I get asked about a lot. Oddly enough, though, I've picked that up more often than I pick milkweed up. And then again, like I said, to that same point, not all bees can collect pollen from every plant. A lot of things in the nightshade family, I know also are buzz pollinated, but I actually pick up quite a few things that are that are in that weedy solanaceous group of plants. Okay, so there's just a couple things to keep in mind. If you see something on your report and you're like, why is that in there? One other thing that I like to tell people too, when you're providing me a sample of honey that's been through an extractor, again, because I'm after the pollen, when you have extracted honey, you've usually collected it in a large volume, right? If the pollen is occurring in pockets in that extracted honey, if it is distributed amongst in a large volume, it can be basically it's diluted, right? It's going to get diluted down. So that's why usually I ask for 2 tubes of extracted honey, because we just know that there's less pollen in it. We like to still offer the option to test that honey, because sometimes, I mean, most people, they're not going to collect like off of an individual frame, right? You're going to wait until you've got enough to bother to put the work into collecting or doing the extraction. And I totally understand that. And most people, again, they want like like what during my peak months of collection, what are the most common plants I'm seeing? I just tell people too, if you're really interested in a particular plant, you may have good reasons to do that frame collection, because you know when that frame has been filled. If you're tracking it. I had somebody that was like, I guarantee you that they're collecting from I can't think of the common name of it. I call them choke cherries. Aronia is the genus name of this plant, but they're like, it's got to be Aronia. It's got to be Aronia. And they gave me an extracted sample, and I was like, well, I didn't find it. And they're like, okay, but I'm pretty sure that that's the majority of what it was. Following year, they did what I recommended, and they did a scraping during when the Aronia was flowering, and I picked it up that time. So it's not that the bees didn't visit it, and that also might be the contributing flavor of what the honey was, and that's what they were trying to verify. But sometimes you do kind of have to, depending on what question you're asking, you might have to cater how you do that collection. And if anybody has any specific questions about that, I treat that as like, sometimes I'll be like, well, collect one sample and then like collect another one and then like send them both back to me and like, I'll just run them both. And that way people like will at least get some type of answer if they're looking to answer a specific question. But I like to bring those things up because again, like any complicated diagnostic method, you're going to run. I mean, not everything's perfect. Let's put it that way. So I just try to make it as good as I can for the individuals that want to use the technology. So again, I am hoping you never ever send me samples for spider lanternfly. As a plant pathologist, there are many reasons I don't want it in California as much as you don't want it in California. I don't want to get more busy at my other job right now. Let's put it that way. So in Pennsylvania, though, obviously we were one of the first states that reported it. And what we found is that honey is an insanely good tool for tracking movement across the state. Because we've had, like, I mean, I don't even know why they're like, oh, this new county is under quarantine. Well, the whole freaking state should be under quarantine at this point. Like Pennsylvania, just give up quarantining counties. We are past that. I have not seen it at my house yet, but I'm pretty sure they're around. So the method for this is very similar to the barcoding, and it uses the same PCR DNA photocopying technique, but there's some modifications to this. It's also faster. Like usually when you give me one of these samples, I can run it within like a week or two. So what the method basically does is, and I didn't develop this method. It was developed by a group at Rutgers that actually designed it for wastewater. So they could, again, environmentally track spotter lanternfly as it moved through New Jersey into Pennsylvania. Anyway, but basically how this works is that when you go through each round of photocopying, which is what these numbers are telling you, it says cycle down here at this bottom, depending on how much DNA of the spotter lanternfly, because that's what this is specifically looking for, the more DNA you have in your sample, the sooner it's going to show up. Like one of these samples looks like it started showing up at cycle 16. And then we have a sample that didn't show up until much farther down in the process at cycle 34. So where my instrument view is able to detect it gives you some idea of the total amount of spotter lanternfly DNA in your sample. But I also tell people I consider this semi-quantitative because I have no idea if in that honey sample it was honeydew or maybe a stupid spotter lanternfly literally fell into the hive. Like that happened one time because we had off the wall numbers and they were like, oh wait, we think it got contaminated with an actual bug. And I was like, well, yeah, that's going to set alarms off pretty high. Interestingly, we can detect it even from the nymphs very, very early in the season. So there's some potential to use honey as a monitoring tool to identify, I mean, because they're going to be within a radius. We mostly know what host they feed on. So if you've got eradication teams, in theory, yes, you could use honey or something else to be able to detect it. But again, hopefully you guys will never, ever have to use this method. All right. So I overviewed the two services that we're currently offering. I kind of joked when I started, I'm like, well, when I first started, we only did the pollen identification with the metabarcoding. And I'm like, well, you really can't call us a lab if we only do one thing. So we probably need to think about offering some additional services that are going to be valuable. Some of those that I'm hoping, the two that I'm going to showcase right now, these I anticipate having the ability to offer them in January 2026. That is assuming that Penn State doesn't hold me up with a lot of paperwork, because universities are not the most efficient things. That's all I'll say about that. But the two services that I'm really looking to offer, and I don't really have a good cost estimate on either of these yet. We're working on making them extremely affordable, like definitely more affordable than the pollen identification. But the first would be a pollen nutritional assessment. This would look at protein, lipid, carbohydrate ratio in pollen samples. And we get a lot of requests for this because, at least in areas like I'm in, where you've got major seasonal changes, it's pretty common for one reason or another that they might supplement and provide more pollen to their bees. We also have a lot of researchers that are looking for that because they're trying to study some of the, many causes that can result in colony collapse. Although I think we all know that most of that's probably being caused by the mites now and the deformed wing virus. But we're getting a lot of requests for just nutritional evaluation of pollen samples for a variety, both from researchers and from beekeepers. The second would be an evaluation of honey characteristics. These would be glucose, fructose, galactose profiles, or fructose profiles, excuse me, and then amylase activities, also one I'm getting a lot of interest in, primarily because like that could be a good indicator of freshness of your honey. And like, again, this is more like just, these are things I'm getting feedback, potentially contaminants, adulteration. That's something we're investigating. We're not quite there yet, but for sure the sugar profiles and the amylase activity, those two I'm hoping to be offer sooner rather than later. And if any of you have ideas about services that you would like to see offered, I'm trying to keep things in the ballpark of what would be valuable to you guys from a marketing standpoint. Like you can say, hey, I know this. I mean, other than obviously like what pollen I can find in that. We can't really do nectar profiling. That's extremely expensive and very cost prohibitive for a variety of reasons I won't get into. But if you guys think of any additional tests, I don't currently do any testing on bees themselves. I'm trying to keep it specific to honey and pollen, as the name says. I may end up going that route of, we've been looking at being able to identify chalk brood, because that can be a real problem occasionally in our area. But currently, because there are other USDA labs that offer that, there are other commercial labs that offer that, I try not to stick my toes in other people's ponds. But if you guys think of any services that are not currently available, let me know. I'm always looking for ways to expand the lab. And I laugh, actually, I didn't say this earlier, but my lab right now consists of me and my 2 technicians. But when I was first running the lab, I joked, I was like, yeah, I'm the manager. It sounds really awesome, except I'm also the only employee. That is not the case now. I have 2 technicians. And yeah, like I'd like to see us get a little busier, like expand things out a little bit. So other resources you guys might be interested in. We've got a new website. I did not have anything to do with this because, again, not tech savvy in that way. But the Center for Pollinator Research has a variety of information that we offer. It's publicly available to everybody. I try to do my part by giving talks. I've given talk. I'm not gonna lie, high school groups are like the most intimidating like groups to talk to. Really fun, but I try to do my part just by getting out and spreading the word, not just about the lab for PR, but about some of the other resources we've got. Okay, so interestingly, a lot of the work that I do, so when you give me a sample, it's given an anonymous name. I don't really know who belongs to what until the very end when I'm sending you your reports. But most of the data that we get, if people are willing to share just their GPS location, because your name would never be associated with any of the data. It's not, the only place it's kept is my private Penn State database, because I have to have someone to track samples. But your information is never made public. It's never, it has no way of getting out there unless you share it. But we do take this information and we use a program called Beescape. I provide some of the, I have not provided any beekeeper client information to them. They've taken all the data that I've generated from our lab and then other researchers that like give approval that yes, you can use this data. But the more information we have about what plants are coming from where, the better Beescape is going to get. Beescape does essentially what you could do with your 2 feet, but on a much bigger scale. It will tell you what your landscape is primarily used for. Is it agricultural? Is it urban landscaping? And it will also to some degree. Obviously, the resolution is amazing in my county. I wonder why. You can look at any place in Center County, you can know almost every plant that came from that spot. But we're trying to work with other researchers to broaden this to, for sure, the continental United States. So hopefully at some point we'll be able to use, now actually what I would love to see some of the historic data go into that. Another resource, I fully appreciate that the pollen identification is really expensive. Obviously some of this is because it's a 4 to 6, or excuse me, it's a 6 to 8 week process, and a lot of that is labor. But I also appreciate that a lot of people don't have the time or the resources to devote to submitting like a gazillion samples to me, or even one. I try to make it as friendly as possible, but another resource that's available for people who do want to try to look at their own honey or pollen samples and identify what they've got. And actually it was really fun. I helped build a good part of this library. I did not take the microscopic pictures, but I did process all the slides that went into the pictures that were taken. And probably like a third of what's in this library came from my yard, because I have a plant problem. Lots of plants. So anyway, but what this website does is that, oops, you can go in and if you are identifying a plant in your yard, you could use it that way. You know, if you did like a photo identification, but you could go on our site and this will show you high resolution microscopic images of your pollen. We tried to make sure that because at first we had just things from the Arboretum and like pretty things from my yard. And I'm like, well, most of what I see bees go to, if like, depending if like you're in a very urban area, a lot of the times it's going to be weeds or like natural plants that are not being cultivated in some way. So we put a lot of effort into trying to represent as many weedy and even, yes, invasive species as we could, because you are very likely to see that no matter where you live. I mean, it's unfortunate, but it's true. And if you're just trying to get it from a perspective of what are my bees visiting, we've tried to populate this with as much as we can. So I'm trying to get as much broad, and I add to this. It's actually kind of funny. I do make my children do labor, and I'm like, go out and find mama some of these weeds, and they're like, okay, great. Mom's weird, but we've tried, and I'm always telling people like, hey, if you've got pollen from this particular plant, can you send it my way? So this is continuing to be built. We encourage, again, citizen scientists to be involved in this as well. Okay, and I apologize if I'm taking forever with some of this. But for all intents and purposes, I've pretty much shared everything I've got. I would be happy to answer any questions. I'm going to go ahead and stop my share and then just go back to the video for anybody who wants to ask me questions. Okay. Oh, yes. Okay, nice. Yes, I have considered that the conifer DNA could be coming from the propolis. I've had people say this to me before. So in terms of the pollen library, if you're looking at a genus perspective, California should be somewhat well represented. I know that quite a few of the species, we have Phacelia. I know we grow California poppy out there. I'd have to look into that specifically and see what we pulled from the Arboretum. I know I grow quite a few California wildflowers because my husband's from Oceanside originally. So he actually grew up in, trying to remember where he wasn't. His parents, my in-laws live in Oceanside now, but he was growing up in the Simi Valley area. So he likes I was like, how many wildflowers did you see in Simi Valley? He did see some. So I try to bring home some California in our yard a little bit too. But again, if people have ideas of more plants, if they peruse the library and have ideas of things that need to be added, I love getting feedback about that. That's an active area that I'm trying to work towards making it better. Because again, that's a tool that if you don't want to pay for the metabarcoding and you have the ability and time to look at your pollen in your honey or your pollen samples, that could end up being a really powerful tool for people that want to put, some time and effort into learning about how to do that. I've been asked about that before. I guess like the terpene analysis, I think that would be fairly straightforward for us to look at. That's been kind of a hit or miss, but that's good that you brought that up because you're not the first person to ask me about that. So I'll put that on my radar and look into that a bit too. We actually have we got new equipment in one of our facilities. I guess I should back up. So my direct supervisor, Dr. Christina Grosinger, I'm part of her lab. And she is now the director of one of our big institutes. And she's been alerting me to all these nice, brand new, fancy pieces of equipment that are hopefully going to make the world, like change the world and make things faster and cheaper. I'm always interested in cheaper, as long as we're not losing quality. And I know at least of a couple pieces of equipment where the terpene analysis might be pretty seamless. And bear in mind, if you send me samples for pollen identification, for the metabarcoding, and you find out later that, yes, I'm now offering that, I keep all of your samples. If you want to ask for another service on a sample, I will keep your sample, and then I'd basically not charge you except for like the very base fee. Because you're not shipping it to me. I'm not paying for that part of it again. So bear in mind, if you ever retroactively know that you sent me a sample and you want to go back and be like, hey, could you do carbohydrate analysis on my sample? Could you do something along those lines? I would be more than happy to do that. All right. Any other questions? Hopefully I didn't bore the pants off you guys, and sorry for the lightning bug interruption.
I had a question, Michelle.
Yeah.
So thinking about, you talked about doing a pollen analysis of specimens collect in historical collections for some kind of historical data on forage. Is it have you done testing of older honey samples, or like, honey samples of 50-year-old honey samples? Is it possible to say, like, so for example, I have a jar of honey from like the 1950s that was my grandfather's honey. Is it possible to send in honey for analysis that's that old, or is there any degradation that happens with the
I've
material?
never done a honey sample that old. I have done wild bees where we pulled off the pollen basket, and we've had pretty reasonable success with that. In theory, it should work fine for a honey sample like that, primarily because one of the main attributes of honey and why it's so popular is it's antimicrobial. Most of the time, DNA is going to break down because of things in the environment that attack basically just DNA that's hanging out there, or will attack the pollen. But because there would be antimicrobial activity in the honey, I would think that there wouldn't be any problem as long as there's enough pollen in there to go after. In theory, it should actually be better than some of these old pollen samples I've got, because when I've run into issues with pollen samples, it's been because they're not always stored at ideal low humidity conditions. And as a plant pathologist, I can flat out tell you, fungi love to eat pollen. I wonder why. So you'll get a lot of bacterial and fungal breakdown of pollen samples that are just left out in the environment. But honey, again, by its very nature, should be antimicrobial. So I would actually think you'd have a better chance of getting historic information potentially off of that than you would just like a pollen sample. That might be the one advantage honey would have over pollen.
That's cool.
Because I mean, I've had people send me fully crystallized samples and they work fine. It doesn't matter. I can't always tell them why their sample crystallized. Like, honestly, I usually am like, I don't know. But I can tell them, like, I know what plants were in there. But yeah, that's a good question. Most people just don't keep honey around long enough for me to really have had encountered that. I did have somebody who had like it was older honey, and they were asking about a specific source though, and I'm like, well, I can't guarantee that. And I think like the price was unfortunately a turn off just for curiosity's sake. which I appreciate. I get that it's not the cheapest. Ah, have you ever tried to detect parasites and pests? Yes, inadvertently. So when we were first working out how to deal with the data analysis pipeline, I had to do a lot of looking at the raw sequence data. And when the DNA is photocopied, part of that photocopying process is you have something called primers, and the primers are what tell you the piece of DNA to photocopy. The primers we use, we know are not exclusive to, they won't just pick up plant DNA, they also will pick up fungal DNA. But we also didn't really care because our data platform will just kick out anything that's not a plant. So it's filtered out. But yes, we know that I've picked up, my favorite so far was I was looking at, we have an apiary that's in the middle of, like, our Penn State lab has an apiary that's in the middle of an agricultural area. And we had corn pollen in there, which, okay, all right, whatever. But we also had a corn pathogen in there that I recognized because that was one of the things I worked on for my PhD. And I was like, holy crap. We don't really use that information right now, but there's been some interest in my lab to go back, particularly for some of our students that are studying solitary bees that have had issues with chalk brood, because a lot of solitary bees get it too. They're like, oh, well, when you pick that up? And I'm like, well, you have to look for the sequence, but yeah, in theory, if it's there, it's a fungus. It should be there if it was there. But believe it or not, data mining, that type of data is sometimes more difficult than just going at the biological approach of just reinterrogating the sample with new primers that are specific for what you're looking at. It's faster, it's cheaper, it's less laborious because somebody still has to be looking at that sequence data. So inadvertently, yes, I have, but we have not, we don't plan in the future to really do that with our data. I kind of wish somebody in my old department and plant path would do that though. I was like, hey, we got all this data. But so far, using pollen has been mostly for viral tracking plant viruses rather than some of the other like fungal or bacterial organisms that we see. Plus, some pathogens don't make it to the pollen, even if they're infecting a plant. So I'm not sure how much you could get a lot of pathologists to buy onto that, but that's a really interesting question. Potentially, we do have that data. But whether or not someone wants to go through it is another matter. And if you guys think of any questions, feel free to contact me too. It might take me a little bit longer in summer. I don't generally work as many hours because I lose some of my student help, but I try to be as responsive as I can.
Thank you. I think I see Tarle has her hand up. Tara?
Oh.
Tarle? I'm not sure if that's an accident or.
No, it's not an accident. Can you hear me?
Yes.
Hi. Yeah, I'm just wondering what's roughly the base pay to get started on the basic package.
So for you guys right now, per sample for the pollen identification, that I believe it's 82.77. You would think that I have to tell people that number, not that I would remember it, but half the time I have to look it up. It's around $85 per sample. If the outside, we usually charge $130 because the university slaps on a fee because of administrative things that I don't really understand, but I'm hoping that actually becomes less expensive sooner rather than later. We have some new technology that I'm hoping to implement where I won't have to change a lot of my methods, but I, in theory, could have the amount of work it takes, which would drop the cost down to more like $50. But again, like that, some of that I have to do the research and look into some of that. So right now it's going to be about 85 a sample for folks. If you do want to submit more than 8 samples and don't want to pay an astronomical amount of money, I'll usually tell people, but like, we can parse it out. Occasionally, if it's like under a threshold, I'll be like, well, I'll throw it in. But I do try to keep it as reasonable as I can. But yeah, for your group, all I need you guys to tell me is that you're part of the Mount Diablo group. I've got you guys tagged under that. I also, if anybody, I'm like, do you watch Penn State games? We could talk about being an alumni discount. I mean, I do the best I can with what people have to work with. Sometimes it just sucks to have to work for like the boss, right, of the university.
Well, thank you once again, Michele, and thank you for the presentation and also for the bulk discounted rate for our club. I know
No problem.
members who have already sent in their samples really enjoyed the process, and it was very easy and very clear for them. So thank you for that, and thank you for taking the time in the evening to come and join us this evening. Thank you very much.
Thank you guys for having me. Again, I love working with beekeeper groups. One of the things I missed most when I wasn't working is extension interaction, and this has been great for me. If you guys know local high school groups in your areas, anything like that, I try to slip them in under the radar. And I might be looking for test users to send me some samples in for some of the new stuff that we're looking at. So keep me in mind. Happy to work with everybody. I apologize again if things are a little slower in summer. 99% of the emails go through me, and like 2 days a week, I'm also stay-at-home mom. So, usually things pick up a little bit, closer to fall, but I will get samples out the door as fast as I can. Thank you again for having me. It was wonderful to kind of virtually meet everybody. I'm sure you're having better weather than I am. If it's 85 here, it's also 900% humidity. So that's kind of sad.
Yes.
But you guys have a great evening, and please contact me if you have any additional questions.
Thank you very much.
Thank you. Have a great evening.
You too.